welcome back to the lecture series in animal physiology. So, we are into the 5th week and we have done with the first class where we talked about the overall idea of excitability, and I told you that most of the cells in our body have a membrane potential of around minus 80 millivolt, minus 70, minus 80 millivolt, barring aside from cells which are sitting at minus 40 millivolt. And with respect to outside, the inside is more negative. Okay? So, in other words, there are more negatively charged species inside the cell as compared to outside. And further we highlighted the fact that outside the cell, if you remember the slide what we talked or what you drew, if you just let us go to the slide, if you remember this slide from the last class. So, if you follow this slide, this is how it looks like. All those blue color spaces what you see between the cells, look, I am trying to again further extend that drawing. So, these circles which are here, right, these circles are the individual cells forming a tissue like mass, and in between these structures is that blue shades, and that blue shading is nothing but extracellular matrix, and extracellular fluid is accompanied in that. So, each one of these different cells are kind of different receptors, they are cross talking with each other, it is kind of a colony. Okay. In between the colony, just imagine that there is a small like road like thing or kind of connector like thing which are filled with fluid and different other extracellular matrix proteins. Okay. So, this is what we call as extracellular space containing I am just putting extracellular matrix here. EC stand for extracellular, M stand for matrix, and extracellular matrix contain extracellular fluid, ECF, F stands for fluid. Okay. So, you have extracellular space which has extracellular matrix which is composed of proteins and carbohydrates mostly, okay, and few other different molecules, and that extracellular matrix contain the ECF, and ECF is, as I have mentioned here, has a large proportion of sodium and a large proportion of chloride which is at a higher concentration, Cl minus, Na plus, okay, and few other positively charged ions. And outside this thing, if you remember, we put a voltmeter across this membrane, like this is one probe of the voltmeter, this is the other probe of the voltmeter, and across this we are measuring the voltage. What we found out is, with respect to inside to outside or with respect to outside to inside, inside is more negative. So, now these minus signs are showing what I meant by that, and with respect to outside it is more positive, right. Now, if you see the membrane as such, then it looks like a part of the membrane will be something like this. Positive, positive, these are the positive charges outside, and here you are having the negative charges. And now, if you show this picture to any person with basic knowledge of electricity and electronic devices, or whatever you have studied in class 8, class 9, class 10, class 11 and 12, this structure is very similar to the component like this, which is called a capacitor. and capacitor is a device which could store charges, okay, which store charges, and capacitor can store charges as well as it can discharge the charges, and in between the capacitor out here, the region where I am putting the dots, it is filled with dielectric medium. Dielectric medium, and it attains charges like this, very similar to it. So, our membrane, the closest analogy of our membrane is, it is like a capacitor. Across its membrane, it can store charges. So, now if you go back to the previous diagram, so each one of these individual cells, you can treat them as capacitor membranes. So, historically what was observed at one point of time, say for example, now I just treat it like, and when a membrane is treated as component of electrical circuit, this is called a equivalent circuit model of the membrane. It's called equivalent circuit model of the cell membrane. Fine. Now, there was something which was very interesting which was observed. The observation was this: if across this membrane, so we told you, across this membrane all the cells are sitting at, with respect to inside is negative, they are sitting at minus 80 millivolt. So, if your y axis on this drawing, if I put the y axis like this, okay. So, if this is your voltage, negative voltage, and out here is the positive voltage, and here you have the origin which is 0, then most of the cells, so if this is 20, this is 40, 60, this is 80, so these are all plus, okay, and voltage is in millivolt, okay, 20, 40, 60, 80. On the negative side, minus 20, minus 40, minus 60, minus 80. These are all again millivolt. Fine. Now, most of the cells have their resting membrane potential or RMP sitting here, or some of them sitting here, and yet there are which sits here. We will talk about this, these kind of cells which are sitting at minus 40 later. At this point, we will only talk about the cells which are sitting at minus 60 or minus 80 millivolt. So, this is, if you across this membrane, if you place a voltmeter, this is what you are going to measure. They are sitting at minus 80 millivolt or minus 60 millivolt. But it was observed that at certain points while recording, there was something which was very unusual. In other words, what they observed was, all of a sudden from sitting from minus 80, this shoots up like this, and it further goes down. This is just a simple recording across the membrane of a cell. So, what people observe, that all of a sudden, and of course, this x axis is your time axis with respect to time, could be in millisecond mostly. Okay. This is the voltage of the membrane across the cell. Some cells all of a sudden shoots up an electrical activity which almost reaches 0 and then overshoots the 0 and comes back to its ground state, but then it further went down. So, here just before I get further, so cells remain is called, they remain in a polarized state. This is a word which will you come across in any neurobiology text or neurophysiology text. The cells are polarized. That means that there is a polarity across the cell, and I think after looking at this diagram, it is clear why it is said the cells are polarized, because there is a polarity across it. So, one pole is positive pole, the other pole is negative pole, fine. That is why the cells are considered as the polar object out there. Okay. So, these polar objects all of a sudden gets depolarized. This is the depolarized state where all of a sudden their polarization is being removed. They became depolarized, and not only they become depolarized, they further go up, became they change their polarity for a while, okay? Because going up like this means what is happening, all of a sudden this membrane is behaving, a part of the membrane on this membrane is behaving with respect to time. Here if I draw it like this, with respect to time, at a certain point this is becoming like this, right? Am I right? Because at this stage when you are measuring out here, look at it, out here when you are measuring, you are saying with respect to outside, the inside is negative, and you measure minus 80 millivolt, right. Then at some unique point you see across it when you are measuring, this becomes say plus 10 millivolt. It means the inside with the same polarity, the inside becomes more positive as compared to the outside. The outside transiently become negative. That can only happen, only can happen if for a very transient period of time, for a very transient, now mark my words very carefully, for a very transient period of time, from this sea of positively charged carriers, a huge population of positively charged carrier drift on this side very transiently, or there is another option, inside this there are some structure which store so many positive charge that all of a sudden all those positive charge comes out. But there is no such structure. There is one structure, but that is a very small structure. Even if it gives out all of it, it is not going to really make the whole membrane positive. So, only other likely option in this game is this: huge amount of positive charges like a drift, like a wave. Okay, I am drawing the wave for you guys to kind of remember, like a wave kind of comes inside, and that comes at a very, so a positive charge flux drifting or diffusing inside the cell very fast. Why it is very fast? The reason why it is very fast is here. If you follow this drawing, I am shading a part, like look at this, one second, let me. look at this. The period of time is, this is the only period of time in which you have to halve that period, because it is half when it is coming down. Okay. So, in other words, what does that mean? Let me put a dotted line. So, this is the only part when it was positive, rest it is coming down. So, there is a positive charge flux diffusing inside the cell at a very, let me add one more very fast rate, as if all of a sudden lot of positive charge is zoom inside it, like all of a sudden the flux in just like a wave, they came boom inside it, and so much so for a very transient period of time the inside of the membrane become over to way too positive. But the very next moment, as if the cell senses that I have two, all of a sudden my whole thing went like, haywire. All of a sudden I became positive. I am not supposed to be positive, I am supposed to be negative. The very next moment, they, as if they, so the next thing what you see, next thing if I had to draw is, it comes back to this situation. So, original situation of minus 80 millivolt. So, it means that those huge number of positive charges which entered there, this was correlating with this diagram out here. And then the third in between something happened, which I am showing in blue now out here. Okay. What is happening? A wave of positive charges again drifts out of the cell. Okay. So, first what we observe, positive charges out here, positive charges flux diffuse inside the cell, and it happened at a very fast rate within a very small span of time, which is shown here. So much so is become plus 10 or plus 20 or whatever, Of course, in my diagram it all the way shoots up, but it generally does not happen, it kind of shoots up to this, okay. Because I did not draw it with scale. So, generally it shoots up something like this most of the time. Okay. So, this is the kind of shooting up we are talking about. It becomes, the cell all of a sudden become. So, if this is the polarized state of cell, then this is your depolarized state of cell, where, so in between a situation comes, before this one is arrived, the situation comes where possibly while moving through, which is this zone of the curve, where across the membrane positive, negative, positive, negative, positive, negative, positive, negative. So, if you put a voltmeter across it, it becomes 0 millivolt. So, there are equal amount of charges in between the membrane, or across the membrane, I am sorry, across the membrane. So, for a transient period of time, the whole membrane becomes depolarized, and then it becomes, out here it becomes positively polarized, right, again for a transient period of time, which is shown by this situation. And out here what you see is the depolarized state where it reaches the 0. Okay? Then the system again falls back to its ground state and it further overshoots the depolarized state out here. If you look at it, it went below minus 80. This zone is as if inside it has become way more negative, more negative than normal. For again, for a very transient period of time, this zone is called hyperpolarized. So, for your understanding, you can put it like that. Initially, the membrane always sits at negatively polarized state. Of course, when we say negatively polarized state, what we meant by that is inside is negative as compared to outside. Okay, this is just an axiom, how we explain something with respect to something. We always have to have a comparison or a data, like a reference point, a reference datum. So, this is the reference. Okay. So, with this respect, this is like this. Okay. So, that is how we explain it. So, you can, for your understanding, you can kind of visualize it as the membrane is negatively polarized state most all the time. All our cells are at negatively polarized state, but some cells, if you remember the previous slide, some cells which are called the excitable cells behave differently as compared to their counterpart which are non-excitable cells. Non-excitable cells all their lifetime maintain this value minus 80, whereas contrary to this, excitable cells very transiently changes their polarity. They first became depolarized here, this is the depolarized, followed by that they become positively polarized and then they again come back like this and then they become hyperpolarized, negatively hyperpolarized and then come back to their ground state. Now, this distance what you observe of going up and coming down, this gap or this time varies between cell to cell, cell to cell. There will be action potentials what we will be dealing later while we will talk about the cardiac system. You may see this gap even goes further like this. In other words, the way it is falling will be something like this, or you will have cells where it may go even further up where you will see something like this, or you will have cells where you see it shrinks down further like this. So, almost what you see is something like this, almost something like this, almost. Okay. So, this going up and falling down is something which tells a story. What it tells? It tells the story that what is the rate of charge discharge, and this word I am using, the rate of charge and discharge from the point of view of the capacitor. If we treat it as a capacitor, so the rate at which this charging and discharging happens. So, if from a layman perspective, if you think of it, if it charges fast, yet it can hold the charge for a prolonged period of time to discharge, that is something what we see in structures like battery or a cell, right, where you charge it and it prolongs over a period of time. You can use a triple A cell or double A cell or likewise for a prolonged period of time. On the contrary, there are devices, there are charge storage systems where, like they charge very high and they discharge very fast, something like this, charge, discharge. Those are something like supercapacitors. Okay. So, exactly on the same way, and yet there are some which are pure capacitors, where they charge and then they discharge at a certain rate. Okay. So, based on these charge discharge properties, one can classify the membranes, what will be their charge storage ability, and that in a way tells, in order to bring out such an act, which is a very, always remember, these are exceptionally, these kind of motions are exceptionally, motion or flux of ion is energy intensive processes, and all these energy intensive processes requires large amount of ATP molecules, NADP molecules, these are the energy currencies, ATP, GTP and all those things which are needed out here. But mostly of course we will talking about ATPs, adenosine triphosphate, where every time one phosphorus group is detached from 3-phosphate, so it liberates a huge amount of energy. So, all these processes are amazingly energy intensive processes, and depending on the charge discharge rate, one can get a fair idea about how much energy will be consumed while these particular cell type or that particular cell type is undergoing this charge discharge process. So, now while we are at the fag end of the class, it brings us to some very interesting aspects. It means the first thing what we learned today is there is a huge amount of flux of positively charged ion inside when the action potential is happening. And if you look at outside, which is dominated by sodium, positively charged sodium ions, so most likelihood, in all likelihood, the only element or an ion which can come inside the cell transiently will be sodium. Okay, will come. I mean, just we are kind of developing the story in a systematic way. But then what we observe is that there is a large amount of positively charged ions which goes out. Are those sodium or are those something else? And how a cell restore its balance over a period of time, that where all the time it has very, very low sodium inside as compared to very high sodium inside. So, in the next class we will talk about what dictates this sodium flux and what is the next ion which goes out and how this whole balance is being restored in a cell. So, with this I will close in today's lecture. Idea is to go slowly and to give you a very clear understanding about the events which happens in the excitable cell. If these fundamentals are once clear in your brain, then you can develop the story. I have full faith on you that you can develop the story on your own. Thank you.